Is this working?
Yes, yes.
Yes. Good morning, everyone, and welcome to the Ophthalmology Research Seminar Series at the University of California, Irvine, Janssen Center for Translational Vision Research. So one announcement, as usually, if you have a question or comment, just chat yes to me, and you will be ready to ask your question or come and say your comment when prompted at the end of the talk. So let's begin, and Kelly will introduce our speaker today.
Good morning, I'm Kelly Yin, a first-year graduate student in Physiological Optics and Vision Lab. I'm honored and grateful to be mentored by Dr. Shirley Yin. It's my pleasure today to introduce our speaker, Dr. Yiannis Koutalos, professor in the Department of Ophthalmology at the Medical University of South Carolina. He is also the Barbara and Stanley Andre Endowed Chair for Bioengineering and Vision Research. Dr. Koutalos trained in physics at the University of Athens and completed his PhD in biophysics at the University of Illinois at Urbana-Champaign, followed by his postdoctoral work at Johns Hopkins. His research has been dedicated to understanding the biology and biophysics of vision, with pioneering work on photoreceptors, phototransductions, and retinal metabolism. A central theme of Dr. Koutalos' research is how photoreceptors handle and process retinoids. Retinoids are vitamin A derivatives that make up chromophores of visual pigments. His studies show that the metabolism of retinoids produces byproducts such as lymphofuscin that are important in disease like age-related macular degeneration and Stargardt disease. In recent years, Dr. Koutalos and his colleague have advanced the work with studies on biochemical phase of all transretinal in human rods and cones. He has also looked at cyclic GMP and calcium-regulating photoreceptor sensitivity, at oxidative stress, NADPH generation, and lipid metabolism in inherited retinal disorders. By bridging molecular mechanisms with disease processes and potential therapy, his work is really a bench-to-bedside story. Dr. Koutalos has received numerous awards in recognition of his scientific contributions. He has published widely, and with more than 4,000 citations, his work has a significant impact on the field. Beyond his research, he has served on NIH and NSF study sections and has demonstrated an outstanding record of securing major funds from NIH, industry partners, and international agencies. What is even more notable is that Dr. Koutalos has been repeatedly recognized for his excellence in teaching and mentorship. He has trained over 20 students, many of them have gone to win awards for themselves. He is a perfect example of a prolific scientist who cares equally about education. Today, we're honored to have Dr. Koutalos speak to us about the fate of all transretinal in human photoreceptors. Without further ado, please join me in welcoming Dr. Yiannis Koutalos.
Thank you. Thank you for the very nice introduction. And thank you for the invitation. It's a great honor and great pleasure to be here. And you can see what I will be talking about. And just to say who are the people who have actually done this work over the years. It's really the work by Nick Boyer, Chun-He Chen, and Lee Adler in my lab, and under a lot of support and encouragement by Rosalie Crouch at the Medical University. And for the work that I will be presenting today, collaboration with Federico Gonzalez Fernandez, who is now at the University of Mississippi, was crucial. He was providing us with interphotoreceptor retinoid binding protein that is actually very important for several of the experiments that you'll be seeing. And I should point out that the work really on human cells was spearheaded by Chun-He. She actually did that against my advice. And she actually showed that it can be done, and she actually set up and developed the whole system. And support by the National Eye Institute and the Barbara and Stanley Andre Endowment. So what I will be talking about today is about reactions that take place in the outer segments of the photoreceptors. This is a cross-section of a human retina, and the outer segments of the photoreceptors are over here. And adjacent to them, the retinal pigment epithelium. And these are going to be the photoreceptor cells, inner segments, nuclei, and the synaptic terminals. And light comes in this direction. So let's focus on the outer segments and the retinal pigment epithelium. And the reactions actually relate to the visual cycle. And there is also the retina visual cycle that I will be talking about later. And specifically now for the rods, the visual pigment rhodopsin resides the outer segments and is actually the light detector. So this is what detects light. Light is the chromophore, 11-cis, retinal chromophore in the visual pigment, makes it into all trans, and this actually generates the active intermediate that will start the biochemical reactions that will lead to a change in membrane potential. So this is the conversion of light to the electrical signal. And that's how we see basically the electrical signal will eventually be transmitted to the brain. So this isomerization that is necessary for the detection of light destroys the visual pigment. And the old transform is active but cannot detect new light that will come in. So you need to regenerate the pigment so you can continue seeing. And the first thing that happens is all trans retinal is released and leaves behind the apoprotein opsin. So the pocket now for the chromophore is empty. Fresh 11-cis retinal comes in, combined with opsin to regenerate the pigment. So this is important that all trans retinal has to leave, right? So that we can go on with pigment regeneration. All trans retinal then is reduced by retinol dehydrogenase 8 using an ADPH to all trans retinol. And all trans retinol now can leave the cell, is transported by interphotoreceptor retinoid binding protein to retinal pigment epithelium, where it is recycled to reform 11-cis retinal. And all trans retinol comes, of course, from the circulation as well to make 11-cis retinal. And this is transferred to the photoreceptor outer segments for the regeneration of pigment. And this is the classical visual cycle, if you like, that is specific for rods, and there is something different going on for cones that I will be discussing later. Now, so why do we care about what happens to all trans retinal? This is the title of my talk, right? And what's the fate? And so these are the reactions that I pointed out before: rhodopsin, photoisomerization, and release of all trans retinal. So the first thing that is important here is that with the release of all trans retinal, opsin is left behind. So the pocket is empty, so it's available for regeneration. All trans retinal has to leave. Otherwise, we cannot regenerate the pigment, we cannot see. The second relevant thing is toxicity. All trans retinal is a reactive aldehyde. It is also a photosensitizer, will absorb light, generate free radicals, and this will actually damage the lipids, damage cellular components, and so on. And also, it will form bisretinoid adducts. And these are also cytotoxic on their own count. And these bisretinoids actually will accumulate as part of lipofuscin in the retinal pigment epithelium. So this toxicity of all trans retinal by itself as a reactive aldehyde, as well as through the generation of the bisretinoids, is actually considered an important factor in the mechanisms for retinal degenerations. And work actually from Chris and Philip and the groups actually have shown that if you actually go and trap the aldehyde in some ways or modulate the generation of the lipofuscin accumulation, actually this is protective against degenerations, against the damage. So what happens to the retinal actually is quite important when we think about retinal degenerations. And then the final step, the reduction to all trans retinol moves the chromophore onwards for recycling so that it can actually make fresh. So these are the three aspects, if you like, for why the fate of all trans retinal is something to consider about, why we would care about it. So how do we go about looking at it? So this is photoisomerization, release, reduction, and the emptying of the pocket so that opsin is available. And the main thing that my lab has been doing actually is looking at the formation of all transretinol in single cells. So the technology that we use actually are looking at single cell, what happens to these reactions at that level. And the main focus has been all transretinol. However, all transretinal can make the bisretinoid adducts lipofuscin fluorophore precursors. These adducts, these bisretinoids will end up as in the retinal pigment epithelium. And in order to study this at the single cell level, what we take advantage of is that all these three actors actually fluoresce. We can actually pick them up at the single cell level. And over the years, we started actually by focusing on all transretinol. This is the fluorescence that we have been actually monitoring mostly. But then later on, we moved to actually look at these actors as well. So first, looking at all transretinol, and with the same setup, with the same approach, we will look at the other ones as well. So this is the way that we do fluorescence imaging of single photoreceptors. Take a retina, we chop it up, we actually get an isolated cell in a dish on a microscope stage, and we have a light source for retinol imaging. We use 360 nanometer excitation light, reflected, focused by the objective lens on the cell. The emitted fluorescence is collected by the lens, goes through the dichroic mirror, and we actually pick up the emission longer than 420 nanometers with a CCD camera, and you can see the fluorescence image here. And this is really retinol, as I will be talking about in the rest of the talk. So this is basically the basic setup. Put cells on an epifluorescence microscope and look at the fluorophores in different ways. And all of the experiments that I will be talking about are done at 37 degrees, and something because it's mammalian cells, it's human cells. This is a mouse cell. So before I move on to human, I want to actually go over one slide with data from the mouse because are going to be relevant actually to what I will be talking about. So this is actually an experiment with an isolated mouse, rod photoreceptor. All the scale bars that you will be seeing are 5 microns. Outer segment, this is an infrared image. And this is from a dark adapted cell, dark adapted animal. We get a dark adapted cell. This is the very first fluorescence image we get. And then we bleach 1 minute of light, long wavelength light. We bleach rhodopsin, and immediately after we take an image, after we finish the bleaching, and then 10, 20 minutes, 30 minutes, 60 minutes, and you can see the increasing fluorescence in the outer segment. And I will be referring to this fluorescence as retinol fluorescence. Take my word, it's retinol. I will show you the evidence down the road, actually, that this is indeed retinol. And you can call it outer segment fluorescence for the time being, if you like. We can measure this fluorescence. We can plot it as a function of time. And the rise in fluorescence that you see, we can actually fit it as a first order process. And this gives us the kinetics, the rate constant for all transretinol formation, 0.06 per minute in the mouse. Now, fluorescence, how do I know what I'm looking at? And we have actually done extractions. We take isolated retinas, so they have been removed from the eye cup. There is no retinal pigment epithelium next to them. They're dark adapted in a dish at 37 degrees. The same experiment that you see on the left, but with a retina now. And we actually stop the reactions, extract, and look at the retinoids. And this is the dark state. This is 11-cis retinal. These are the oximes, because we use hydroxylamine to stop the reactions. And this is 11-cis retinal in the dark. We bleach immediately afterwards. We look at what's going on. All transretinal now. And retinol has started forming. This is the peak for retinol. And as time goes by, the peak in retinal increases, and the peak of retinal goes down over 90 minutes. We can put together, because we have the absorbance for the different species here, and we can actually quantify what we have. We can pull all this together, and then we can plot of the retinal that has been released, what is the fraction that has been converted to retinol? And this is what this plots here. And the fraction actually increases with time. The rate constant, again, first order, we fit this with a first order rate constant. It's 0.037 per minute, plus similar actually to what we're getting from the isolated cells. And a very important number here, the fraction that you see at long times after bleach, after all transretinal has been released, all of it has been released from the activated rhodopsin, is 0.8. It's about 80%. So at long times, you reach a state of equilibrium, a steady state, where 80% of the released chromophore is the transretinol. And this is a number, keep it in the back of your mind, actually, I'll come back to this. So this is mouse. Now we're moving to human. After we actually practiced, we graduated, and June had graduated and then taught the rest of us. And these are actually now experiments done with human cells. And this is actually what you see, it's an isolated retina from a human cadaver eye. And this is the optic nerve part. You can see the macula, you can see the spot, the arcades. And from this retina, we can isolate cells, photoreceptor cells, a rod and a cone shown here. And this is actually why I did not believe that this could be done, because this is actually takes at least a day to reach us after death, but it can actually be done. And we actually get plenty, at least plenty of photoreceptor cells that are alive to work with. So we can now go ahead and do the same experiment that we did with the mouse and do it with human. So this is an isolated human photoreceptor, and we have not regenerated anything. And just humor me for a minute. So this is a dark adapted, if you like. We have taken the eye, dissected the eye in the dark, and so on and so forth, isolated the retina, chop up, get an isolated cell. This is a human rod. We bleach, it's a dark state, we bleach, and we can see actually a fluorescence increase with time, similar to what we have seen in the mouse. When we plot, you can see, actually, you can tell it's faster, and when we plot actually the fluorescence, the outer segment fluorescence, it is indeed faster. The first order rate constant is 0.35 per minute. In the mouse, it was 0.06 per minute, right? And you can say, well, these are not going to be dark adapted, right? And of course, they're bleached. And so this is actually, we were fortunate to be able to get cells that had enough rhodopsin that we could do this experiment. Typically, that was not the case. So that we could not get cells that had any pigment. And if you do not have the pigment, you do not know what the problem might be. The cells might be dead. But we were able to get a few cells where we could actually get this, so that yes, it can be done. The cells are alive. They can actually generate retinol. And then what we did, actually, we regenerated the isolated cells using 11-cis. We need to use IRBP, interphotoreceptor retinoid binding protein, as a carrier to do that. Other carriers do not work well with us, at least. But regenerating with 11-cis retinal, we can actually do more experiments like this. Again, isolated rod, regenerated with 11-cis in the dark, right? And then we do the same experiment, and you can see the fluorescence increase as well. And this is plotting the fluorescence change over time after bleach. And the rate constant here is 0.17 per minute. Now, I've done these experiments with cells from different donors, and I'm going to put the so this is the formation rate constant, how fast retinol is forming in the outer segments of the photoreceptor cells. And these are from the unregenerated cells. We had 3 donors that gave us cells that we could actually measure the formation rate constant. We had enough fluorescence to do it. And these are from 3 other donors that we regenerated the cells with 11-cis retinal. And when we pull everything together, there wasn't much difference between regenerated and regenerated. That was important, that they're actually consistent, that the overall mean was 0.4 per minute, 0.4 per minute. And this is from a total of 44 cells. And remind you, mouse was 0.06 per minute. So human is much faster. And this is actually very important, as you will see. So go back to the visual cycle, regeneration of the visual pigment of rhodopsin, right? And this is the reaction that we're looking at, the formation of all transretinol. And I will switch from rate constant for formation to half time for formation, and I'll explain why in a moment. So when you convert the rate constant to half time, you get about 1.7 minute half time for the formation of all transretinol. And this means that opsin is going to empty faster than 1.7 minutes half time. And the chromophore will be ready for recycling within 1.7 minutes half time. And what we do know is that the half time for the regeneration of rhodopsin is about 6 minutes. And this is the issue here, that the regeneration process for rhodopsin is not a first order reaction. It goes up as a ramp, actually. And this is the number that I have is from a review of 2004 by Lam and Pugh, where I actually put all this together and gave these very helpful numbers. And this time here actually is much faster, or sufficiently faster, to allow for this regeneration to occur. If you look at the mouse, the 0.06 per minute, this half time would have been 11 minutes, which is too slow to account for this. In 129 mice, which have the RP65 with leucine at the 450, and they're reasonably fast at the regeneration of the pigment, this half time is 20 minutes. So there is actually a matching, if you like, or these numbers move together. So this is generally faster than this one, which makes sense. And mouse is slow compared to human. We regenerate, primates regenerate fast compared to other mammals. So, okay. How about cones? And this is a story of blood, sweat, and tears. We were able to get some cones, and we had to regenerate. We could not get any cones that we could actually get a signal without regeneration from human. And we did get a time constant for formation, exactly the same way that the experiment is done. We bleach for 15 seconds with white light, and you can see the fluorescence increase in the outer segment of the cone. And the rate constant for formation is 1.8. And we can have a discussion at the end, and how can I trust an n equal 3 for the 1.8 per minute, but there is an answer to that. So now we go to Vladimir's work. Basically, he demonstrated very, very nicely that for cones, the regeneration of the visual pigment involves the retina and a pathway that involves the Miller cells. And here is the photopigment, the cone photopigment with 11 cis retinal. Again, all trans, the active intermediate is formed, all trans retinal is released, leaving behind opsin. And again, reduced, all trans retinol. This one now is going to go to the Miller cells and be made into 11 cis retinol that is going to come back and make 11 cis retinal for the regeneration. And the numbers here, the half time is 0.4 minutes. And the half time for the regeneration for the cones is about 1 minute. Again, the emptying of the pocket, right, is fast, and the recycling is fast enough to allow for the fast regeneration of the cone pigment in the human as well. Okay. So this is the basic experiment, right? Dark adapted photoreceptor, bleach, fluorescence increases, goes away, and these are the reactions that we're looking at. So this I have been doing for many years. And then at a FASEB conference, 20 years ago, I don't remember, Chris. Chris actually pointed out to me that all transretinal also fluoresces. Oh, okay, yeah, but it doesn't, right? Not very much, at least. But how do I know? So basically, how much of this fluorescence is all transretinol? How much could be contributed by all transretinal? Because these are the two retinoids that you're looking at. So I followed the advice to look at all transretinal. And because it fluoresces, we can actually look at it as well. And because all transretinal and all transretinol have different excitation spectra, we can actually separate the signals. So these are absorption spectra for all transretinal and all transretinol. All transretinol peaks at 325 nanometers, all transretinal at 380. This is an equivalent in ethanol. The isosbestic point here is 340 nanometers. So what we do is that we can excite at 340 and at 380 nanometers and look at the emitted fluorescence above 420. So this is a broken off rod outer segment, human. And this we load with all transretinol for 5 minutes. And we excite 340, 380. 340, much higher than 380. Retinol is much brighter than retinal. We load the same broken off outer segment with retinal. You can see that 380 is much larger than 340. So 340, 380 for retinol, 340, 380 for retinal. Goes the opposite direction. We can actually measure the ratios, 340 over 380, and these are actually for retinal, and these are for retinol. So it gives us a nice range. So if we have a mix of the two, so the ratio is going to be in between, and this is basically using this as a ratiometric die. And we can actually do the numbers, and we have a formula actually that for the fraction of retinol that is present is given by this. The 1.9 is the inverse of the ratio for retinal. The 0.16 is the inverse of the ratio for retinol. And Q here is the ratio of the fluorescent quantum yields for retinal and retinol that we have measured independently. And it's 5 to 15. Retinol is much brighter than retinal. Now we can use this to go to a cell and look at the experiments with the cells. How much is retinol? How much is retinal? So first with unregenerated human rods. So this is the endogenous retinal being released. This is the dark. We bleach and we measure immediately afterwards, and then at 10 minutes after light. 10 minutes is close to the peak, basically. That's why we picked up 10 minutes, close to the peak of fluorescence that appears in the outer segment. Again, you can see when we show this, it's retinol, much brighter at 340 than 380. You can actually get the ratios, and it's 5, 6. We then can do it with regenerated rods. Again, nicely, looks like retinol. And again, the ratios actually agree that actually are fairly high. And we can do it with cones as well. You can see the cone outer segment here. And it's 1 minute after light. Cones are fast. So we're looking at them pretty fast, fast after the bleaching. And this is actually, these are the ratios for cones. And all this is very heartening. What is also important, we can actually plug it in the equation, see its fraction of retinol. And 0.8 to 0.9. That's why you have kept in the back of your mind what was in the mouse from biochemistry, which is about 80% fraction of retinol. And when we had done the same experiments in the mouse, we get the same 90% conversion. Okay? So most of the fluorescence that we see, if we include the brightness and so on and so forth, is going to be 99.5% of the fluorescence in the outer segment is actually retinol. So we have been looking at all transretinal, and because with the difficulty with the human cells, because they do not always have rhodopsin or enough rhodopsin to experiment, right, to do a meaningful experiment, we actually had to devise a different approach to actually look at how they handle all transretinal, again, for the reasons that I discussed in the beginning. Because of the toxicity, we were very interested, how are the cells handling all transretinal? And what we actually developed was a system where we can actually load the cells with all transretinal from the outside, provide all transretinal. So we bleach the cells, so we don't have to worry about endogenous chromophores. So this is another segment. We add all transretinal with a carrier, bovine serum albumin. It's converted to all transretinol. It uses an ADPH. All transretinol leaves an ADPH, continuously cycling. The cell is continuously generating an ADPH to reduce retinal to retinol. And this is a photoreceptor, human, outer segment, inner segment. The enzymatic machinery that generates the NADPH is in the inner segment. And the way we do the experiment, we take an initial measurement, 340, 380. We load, we bathe the cell with all transretinal, 5 micromolar is our standard protocol for this. And we wait for 5 minutes. We take another set of pictures, 340, 380. We subtract, and we have the ratio 340 over 380 for how much of the retinal that we added, right, that is loaded on the outer segment has been converted to all transretinol. And since we're getting the ratio from this, but the other thing that we can get is the absolute intensity excited by 340 nanometers. So on the left, we can now compare the ratios, 340, 380, this is the pure retinol. This is from the endogenous, what I was telling you about before, right? Or endogenously released. This is actually these are the ratios that we get with the exogenous. And they're actually pretty nice there in agreement. 80 to 90% with exogenous all transretinal. However, when we look at the intensity of fluorescence excited by 340, which tells you how much retinol we have there, when we get, these are the levels from the endogenous, exogenous are about 5 times higher. So really you're loading the cell with a lot of all transretinal, and the cell is doing very fine. It's actually converting 80%. So it's handling high all transretinal loads very well. We were surprised, but also pleased. Maybe this will be a question down. But the whole idea here is that the capacity of the cell to handle all transretinal is several times higher than what it needs to handle the endogenous loads under physiological conditions. So, okay, if it's handling all transretinal so well, where do you get lipofuscin fluorophore precursors? And can we actually think about it? Can we measure this? And what we did actually, we looked, can we see these precursors in isolated human rods? And yes, we can see them. So we actually take an isolated cell. This is an image of the cell, Nomatsky. Look at the fluorescence. This is actually 490 nanometer excitation, higher than 515 nanometer emission. This is true color. This is actually what you would see through the eyepieces. And you see this very nice orange fluorescence in the outer segment, which looks very much like lipofuscin. And this is what we have been calling lipofuscin fluorophore precursors. And this is, this nice green is most likely FAD. It's in the mitochondria. It's oxidized flavonucleotides. And when we measure the emission spectrum from outer segments, peaks around 600 nanometers. Again, consistent with the fluorescence of lipofuscin granules. So can we actually look at how retinal can generate these kinds of precursors? And we do the same experiment as before. We can add all transretinal from the outside, and this could actually be reduced to all transretinol, and it could also form the precursors. And we can combine the measurements, right? We have a cell sitting. We take a basal, an initial measurement, 340, 380, 490 for the precursors. And these are the basal levels. All transretinal, wait for 5 minutes. We take another picture. We have 340, 380 for the ratio. And you can see here, actually, you can see the precursor fluorescence increasing. So now we can look at the relationship between the reduction of retinal to retinol and formation of precursors. And had 62 cells. This is the basal level of precursors for these cells. And then these are the levels of precursors after the addition of the transretinal. And as you can see, as the ratio goes down, as retinal is less reduced, right? As we have more retinal, the precursor levels go up. So this is not very profound, that if you do not reduce all transretinal, you will actually get a lot of lipofuscin fluorophore precursors. But we actually demonstrated it. And in order to do this, actually, we had to get levels, we had to have cells that do not reduce all transretinal very well. For that, we actually used sicker human cells that we were dissected several days. We actually isolated several days after the initial dissection. So they were going downhill, basically. The retina was going downhill. So actually these down here, instead of looking in a haystack for an unhealthy rod, we actually went for several days after dissection, and we got plenty of unhealthy rods in that case. So we actually were able to get a nice spread actually of values. So this is actually, I found actually an important message here from this, because what I feel is that this actually points to what could actually underlie transretinal toxicity in a chronic fashion. As think actually for how things operate in the eye. So we have photoreceptor cells in relation to the RPE, and if there is a metabolic impairment, the photoreceptor cells will not be able to reduce all transretinal very well, so you will have accumulation of the precursors. This will end up in the retinal pigment epithelium. There's going to be increased lipofuscin accumulation. Increased lipofuscin accumulation will result in lysosomal dysfunction, will actually make the retinal cells unhealthy. They will not be able to support the photoreceptor cells as well. Photoreceptor metabolism will go even worse, and that's actually a spiral. That's a positive feedback loop. What is important here is that you can enter this feedback loop at any point. You could actually have a disease with an enzymatic defect that leads to all transretinal accumulation. This would lead to the lipofuscin accumulation, the cells actually getting sicker, but this sickness, this initial impairment will lead to even worse accumulation, which will eventually lead to death. So I like that this had actually a chronic element, a time element in it. It was not an acute effect. And to actually pull everything together about what I talked about, to summarize what I have discussed today. So this is basically, these are the reactions that take place in the outer segment, the visual pigment, photoisomerization, release of all transretinal and reduction, and all transretinal can also form precursors, and the 11 cis retinal that comes in for regeneration. So one important thing, there is actually 8% conversion of retinal to retinol. And the half time for this formation of retinol is 1.7 minutes in rods. And this actually allows available in less than 1.7 minutes, which allows for regeneration. And a similar thing, conversion time constants hold for cones as well. Another thing is that the metabolic capacity of the cells is plenty to handle the physiological demands. So if the cells are healthy, they can handle all transretinal very, very well. And finally, it's metabolic impairments actually that will lead to the increased formation of lipofuscin precursors. that could, of course, get things in a spiral. I'll finish here. Thank you so much for your attention, and I'm happy to answer any questions.
I'll start with one question on technical level. You're bleaching for 5 minutes or
1 minute for rods is 1 minute.
1 minute. And then you're using fluorescent to detect it. Does this fluorescent light also bleaches continuously pigment?
Yes. So what we have done, we have actually checked whether the fluorophores actually bleach by the excitation light. So we actually had to make sure that we were working at levels that will not destroy that. There is no pigment left to worry about bleaching of the pigment, so that it will have any extra release. And so the way that we bleach the retina for the 1 minute is the same way that we bleach the cell, and we get 95% bleached pigment from these under these conditions.
Philip? Vladimir?
Hey, Yiannis, beautiful talk and very thought-provoking. Thank you. I have many questions I'll ask too, if I may. So the 80% or 90%, so what happens with the remaining 10, 20% all transretinal?
So this is an isolated cell sitting without IRBP around. So it takes time for retinol to leave. If you put IRBP, in the mouse at least, where we have done the experiment, retinol is going away. So basically the reaction is going to go forward through mass action by removing retinol, and then retinal is going to go away. So we have done the experiments with IRBP in human photoreceptors. IRBP takes retinol away. What we have done in the mouse, we have done the experiment with IRBP present from the beginning of the bleach, and the fluorescence is really tiny in that case. Really, it removes it very fast. We cannot do that in human, but it's just,
Okay.
it would have been technically very difficult to actually set it up that way.
And my second question is about the cones. So there was a beautiful paper by Satoru Kawamura and Shinya Sato, who is actually here in the audience, that I'm sure you're aware of, suggesting that the oxidation and reduction are actually coupled in cones. So you have the conversion of all trans retinal to retinol, coupled with the conversion of 11-cis retinol to retinal. So can your measurements actually pick that up? And is this something that you could test experimentally in either mouse or human cones?
So the 11-cis forms of retinol, I don't see their fluorescence. So so I cannot really see.
But you'll need to put all trans retinol, I guess,
So
11-cis retinol to drive the
so
back
we have done
reaction.
experiments with 11-cis retinol, but it's we do not see we do not see 11-cis retinol or 11-cis retinal. 11-cis retinol does regenerate the pigment.
Right.
Yes, and you will say so you will get fluorescence afterwards. So you can regenerate the human cones either with 11-cis retinal or 11-cis retinol. Yes.
But if you put 11-cis retinol, can you see an increase in the fluorescence coming from all trans retinol is what I'm asking. That will be the experiment.
We haven't done that.
Okay, thank you.
Thank you, Yiannis. This is a fantastic talk. Two questions. So, in your measurements, you see a lot of fluorescence appearing in the inner segment. And I was wondering if you said you've done experiments with IRBP around, does that happen with the IRBP present? Does that kind of draw it out, or it's just interesting that there's so much that kind of are going backwards, the sort of the wrong way.
Yes.
So that's first question.
Yeah. So we have checked into that in the mouse. So in the mouse, where we could actually do the experiments in a different way, there is a leak of retinoids from the outer segment to the inner segment. So at the time, we thought also that this is relevant in terms of toxicity, especially if all transretinal moves to the inner segment, and that's where we felt RDH12 would be relevant for detoxification. We see a similar leak in the human. We have not been able to quantify or probe it simply because of the situation with the human cells. When you start regenerating, I do not know, because it's a whole cell sitting in a dish, I'm not confident that we're actually going to deliver the chromophore only to the outer segment, regardless of what the carrier is going to be, whether it's IRBP or bovine serum albumin. For the all transretinal, we never try to regenerate with bovine serum albumin. It doesn't work well. IRBP works beautifully, but we haven't seen a lot of the fluorescence actually coming from the inner segment. There's some, especially if you put all transretinal from the outside, yes, you're going to get fluorescence from the inner segment as well.
This is something I've thought about and wanted your thoughts on it. Sorry, I think this might have. So, in most cells, if we have retinoids going from one membrane to another, there's a carrier protein for this, like, or from one cell to another. But there's not in the outer segment, as far as I know. There's nothing getting this retinoid from the disc membrane to the plasma membrane. And I was wondering what your thoughts are on that. Why we don't need it in that situation?
So several years ago, some people thought that IRBP is irrelevant, even for the transfer between outer segments and RPE. It could be that simply because the distances are so small, actually, it doesn't really matter. Now, retinol will move easily out of the cell. And you can see it actually in the mouse where it has a large surface to volume ratio. It leaves the cell. Retinal is very sticky. It will not leave the cell easily. So the way that I think about is that retinal will not move from one membrane to another. It will have to be reduced. After it's reduced, it will just hop. So retinol will simply hop actually from the disc membrane to the plasma membrane. But retinal has to be reduced, otherwise it's going to stay there.
All right, and we have 3 questions from the Zoom audience. Roxana, are you ready to ask your question?
Yes. Hi, Yiannis.
Hi, Roxana.
An excellent work. So I just wondered, under your experimental condition, when you use that, I guess, excitation at 480, right? Or close, 490.
490, yes.
So that is also close to protonated and red PE. Could you comment about the status of this?
Shift basis. Yes. So
Thank
shift
you.
basis.
Yes.
So, there's gonna many things to say. So we have no way of measuring shift basis, maybe. But the short answer to your question is that yes, what I have been calling lipofuscin fluorescence precursors could very well be shift bases. And I do not know, I've been calling them lipofuscin precursors simply because of the similarity of the emission spectra, and the color with the lipofuscin granules. What we have done, we have actually put the photoreceptors and done in mouse in hydroxylamine, use the concentration and see what happens. And we actually saw 3 populations of fluorophores, if you like. One disappeared immediately. Put the hydroxylamine on, poof, about 30% of the fluorescence went away. Then another 30% or so went away over the course of minutes. And then there was about 30-40% that just stayed there up to 10 minutes. So I would say that something like 40% would not be shift basis in terms of how I think about shift basis. The other 60% could well be shift basis. I think of shift bases as precursors to the lipofuscin, to the bisretinoids. So that's the first step actually for the formation of the bisretinoids. So if there are shift bases actually around this would actually move on to form the bisretinoids. The other thing is that in terms of the overall experiments with when we look at retinol and retinal, when hematologists, Philip and Chris actually, they looked in the release of retinal from membranes, and they actually classified what happens to it. In the absence of NADPH, when you do not have reduction, about 40% of the all trans retinal becomes shift base within less than 10 minutes. But if you have an NADPH, if you have reduction, you don't see it. So there's no shift base formation actually. And this would be consistent with what we see long term in our case, that we do not see at long times actually do not see any residual retinoid that would actually be shift base. But in terms of the fluorophores, the orange fluorophores, this could be shift bases. But I do not know whether in my very simplistic thinking, I cannot exclude it, and I would not exclude it. I'm not sure how much I answer the question.
Yeah, it's good. Thank you. Yeah, because fluorophore, when I look at lipofuscin precursor, you have to have a second aldehyde, so it's a dimer. So those are, in my opinion, more, kind of, whereas the NREDP, it's still reversible reaction. So
yes. And actually, to emphasize this point, what I have found surprising is that the reactions that you're looking at is over the course of minutes. When people have tried to generate bisretinoids in retinas, it takes hours to days. Am I correct? Yes.
Yeah.
So I'm discussing it with Janet and also as well. We do not know what these fluorophores that we're looking at are in terms of whether they're bisretinoids or shift bases.
Okay, thank you, Yiannis.
All right. Gordon, are you ready to ask your question?
Sure. Hi, Yiannis.
Hi, Gordon.
I was really surprised that the conversion from retinal to retinol was so slow, a time constant of more than a minute. But maybe Vladimir answered the question. If you've got IRBP in the medium, then by mass action you're going to speed up that reaction. Yeah. So, do you have any idea under physiological conditions how fast this conversion?
So my sense is that what the time constant that you see, the formation rate constant, reflects the release of all trans retinal from rhodopsin, from photoactivated rhodopsin. And it's not the dehydrogenase reaction. What it looks like in terms of the stability, the relative stability of the 340-380 ratio is that the reaction is at equilibrium. That has been my assumption, actually. And it's consistent with what we know from NADPH levels and so on, and pH. So the conversion of retinal to retinol reaches equilibrium very fast. And it's what you see in terms of time constants is actually the release of retinal and then the removal of retinol from the cell, for which you need IRB.
Do you have any idea how long all trans retinal is free in the outer segment?
So under physiological conditions, in the presence of IRBP, there's going to be very little all trans retinal.
Yeah.
That would be for healthy cells. So that's very important. If there is not enough NADPH, for example, or if there's an enzymatic defect for the clearance, it could accumulate. It could well accumulate. But for a healthy cell, it's just, we don't really see much of it, in the mouse at least. And I would imagine the human actually is going to be even, the case would be even more so because of the higher concentration of IRBP in the interphotoreceptor matrix.
So from all of this, I conclude that the contribution of all trans retinal binding to opsin and stimulating the cascade is a reaction we can forget about.
I would think that this would be a minor contribution to the cascade, actually, for the activation of the cell, for actually adaptation, right? Light adaptation of the cell. Yes.
Okay. Thanks.
All right. This is a good lead to the next question. Kimberly, are you ready?
Yes. Thank you very much for the nice talk. Regarding the human measures, I wonder if you have any data to evaluate whether the photoreceptor all-trans retinal toxicity is the same, whether the opsin gene is a wild type sequence or whether it's an opsin gene allelic variant.
Yeah, I do not know. I haven't thought about it actually. So I would think that where the closest to this question, the closest to this question that my thinking has gone is actually whether the release of all transretinal is too fast, for example, or and in that case, it could be an overload. In terms of my thinking, I would think more of if the regeneration of opsin by 11 cis retinal is too slow, so 11 cis retinal hangs around too long. And then one would worry about 11 cis retinal toxicity in that case.
Is it possible with your methods to actually differentiate the different allelic variants and their contributions to toxicity?
No.
Yeah, it's a measurement difficulty.
Yeah, I understand. No, I don't think so.
Okay.
But I haven't thought, but my initial reaction would be, oops, I don't think we can do that, but who knows? Maybe if we can go back to the drawing board, we can actually figure something out.
Okay, thank you. Very nice talk. Thank you.
So I have a question here.
Yeah.
And did you try actually to look at the fluorescence lifetime? Because they are very different.
Yes, I would have liked to, but no, I haven't. But yes, it's something that I'm very interested in.
It might really provide some
Yes.
additional light on the result.
Yes.
Hi, Dr. Koutalos. Thank you for a wonderful talk. My question is also about the transportation of these chemicals. I'm wondering how does 11 cis retinol firstly it's transported to the outer segment, especially we just mentioned it's quite sticky, and is there any disruptions that may cause impairment of this transportation process?
So all the experiments show that the thinking is actually that it's IRBP that transports 11 cis retinal from the RPE to the photoreceptor outer segment. And we'd imagine the same thing would be for 11 cis retinol to be transported from the Miller cells to the cone outer segment. That would be the thinking in that case. So there is experimental evidence to suggest the IRBP involvement and having IRBP. The IRBP knockout mice, the things are not very clear there, but there are actually a lot of issues with that because there's degeneration, there's a variety of considerations in that case. And the other thing is that also the membranes are close of RPE. The processes actually in the outer segment are close. So it could well be that it hops. So that how much IRBP would be relevant is maybe not as clear. But the thinking is that you need the transport actually. And IRBP definitely facilitates the transport.
Hello. Thank you for today's seminar. Is there a specific threshold or concentration of all trans retinal in the retina that will cause significant enough formation of toxic bisretinoids resulting in impaired vision?
The data that we have is that the cell can handle loads that are actually several times higher than what you expect physiologically. However, one of the experiments that we did is this. So this is actually looking at the exogenous all transretinal. So we're actually loading the cells with all transretinal. And this is the ratio, and this is time. So this is immediately after loading, 5 minutes after loading, and 10 minutes. And these are different concentrations of all transretinal exogenous, 2, 5, 10, and 20 micromolar. And what you see, It's doing well with 2 micromolar and 5 micromolar, the ratio stay high. And this was an important experiment to make sure that 5 micromolar is an okay concentration. But if you go to 10 micromolar or 20 micromolar, the cell cannot handle it. It overloads the metabolic machinery. So in our interpretation of this experiment, that if you have too much all-trans retinal, the cell will not be able to make NADPH fast enough to keep actually taking care of the supply. But this agreed. And data from the monkeys, and similar for rods. So the rate constants for monkeys, for for monkey rods and cones agree reasonably. Actually, they're pretty close to what we get from the from the human. So no, I'm not very good with statistics. I try, but I also prefer to have an experiment. So yeah, so definitely it's a concern. But with a monkey, actually, I felt more comfortable that we actually had a decent handle on that.
Thank you.
A great seminar, great questions, and the final word goes to Caroline.
Hi,
Yes.
I think it's on? Okay. Oh, okay. Hi.
The microphone is here. You don't stick to the wire.
Whoops. Okay. Of course. Hi, thank you so much. It was a wonderful seminar. So my question is something you've alluded earlier. You've collected these single cone photoreceptors from a plethora of species and also conditions. Have you were able to, when you were talking about collecting these later time point photoreceptors that perhaps could allude to metabolic impairments, were you able to collect actually disease states either from donor eyes or perhaps preclinical strains of mice to actually compare and contrast to how the unhealthy, quote unquote,
Yes.
photoreceptors would look like?
No, so yeah, no, it's a great question. This is something we are very interested in. We haven't done that. We actually focused on eyes without diagnosed eye disease. And what we got interested in is going to eyes that have a disease and go to areas with lesions and so on and see what the cells would behave like. We haven't done this. And it's more difficult to get those eyes. So we were able to do a lot of experiments, but we actually had to really open up the protocol, keep it a little bit restricted, but also open it up so you can actually get enough eyes to do experiments.
The speaker.
Thank you.
Thank you for coming.